This is a virus. Viruses infect healthy cells in your body so that they can replicate and spread. But how do we detect a virus, like COVID-19, once it's infected you? To get that answer, we first need to understand two essential parts of the virus. First, inside is the genetic material. This is what allows the virus to make copies of itself once it's infected a cell in your body. On the outside, a live virus has markers, also called proteins, on its surface. Many of these markers are specific to the virus and are not found on other cells in the body. When a virus enters your body, your immune cells can identify the virus through these surface markers. Once the virus is identified, immune cells create antibodies that can attach to the viral markers. These antibody tags then designate the virus for destruction. This process of antibody production, however, can take time. So how do doctors and scientists use this information to detect the COVID-19 virus? There are two main methods. The first method tests for the genetic material in the virus. This method isolates part of the genetic material. Since genetic material is so small, it uses a method called PCR, which makes millions of copies of the piece of genetic material so that it can be easily detected by laboratory machinery. There are three different types of samples that can be tested using this method: a nasal swab, a saliva sample, or a fecal sample. After you are first infected, nasal samples can accurately detect the virus for a shorter amount of time than saliva or fecal samples. These tests are all highly specific, so a positive test means you are very likely to be infected by the virus. The second method for identifying a COVID-19 infection tests for the virus-specific antibodies made by your immune cells. The only way to test for these antibodies is through blood. Your body produces two types of antibodies in response to a COVID-19 infection, IgM and IgG. While they are both produced at the same time, IgM production decreases as the infection resolves, so it is only detectable soon after the infection, whereas IgG production can still be detected long after the infection has left your body. So which test will you be given? This depends on when you became symptomatic, as the ability to detect the virus in different ways changes over time. If you currently show symptoms or have shown symptoms of COVID-19 within the last 3 weeks, PCR tests are more likely to be accurate, whereas antibody tests are more accurate beginning 3 weeks after initial symptoms. In the first few weeks after the onset of symptoms, if you are in fact infected with COVID-19, PCR tests on nasal and saliva samples are the most accurate and likely tests that you will be given. In week 3, PCR tests are still highly effective. However, virus-specific antibodies have now been produced by your immune cells, allowing antibody tests to be used. By week 3, these antibody tests are more likely to give you an accurate positive result than any type of PCR method. By week 5, IgM production begins to decrease as the infection resolves. As a result, an IgG antibody test will be more accurate in testing for COVID-19 positivity than an IgM test. From week 6 and beyond, IgM production is limited, so only IgG antibody tests can be used to see if you had a previous COVID-19 infection. What does this all mean? If you are currently symptomatic or your symptoms started in the last 3 weeks, a PCR test is an effective and accurate method to determine if you are infected with COVID-19. If your first symptoms were more than 3 weeks ago, an antibody test is the most effective and accurate method for determining your COVID-19 infection status.